Well, I think we can start. I'll give you a brief introduction, and then you can take it away. I'm sure we'll get some more people trickling in. All right, so we are recording already, so I don't have to hit record. So hello, everyone. Thanks for attending our monthly IBANGS seminar series. I am Paul Meyer, and I am flying solo today because Anna Del Prado is traveling on vacation. So if there's technical difficulties, I blame my naivete. Today, I'm excited to introduce Dr. Roshan Jain, who's an assistant professor in the Department of Biology at Haverford College in Pennsylvania, just outside of Philadelphia. Dr. Jain is actually from Buffalo, where I'm located now, and was a research intern at the University of Buffalo for a while before going to get his Bachelor of Science from Cornell in 2000, where he actually already began his work in genetics, studying bacterium with James Shapley. And he actually got his first author publication from that undergrad work. That's pretty cool. Then he went to Princeton to work with Dr. Elizabeth Gavis on Drosophila development, and published on the regulation of several developmental genes as part of his dissertation. He got his PhD from there in molecular biology in 2007. After that, he went on to do a postdoc with Michael Granato at Penn, which is where he, by my reading of your CV, Dr. Jain made the transition from sort of molecular genetics to behavioral genetics. And he was very productive during this time, publishing several papers looking at movement and learning phenotypes in zebrafish, using both forward genetics and single gene approaches. Then after his postdoc, he moved to Haverford, and I guess that's not a far move. Maybe it wasn't, maybe you didn't even move. In 2015, where he still is now, continuing his zebrafish work, looking at decision-making and memory phenotypes in larval zebrafish, and has been developing and studying zebrafish mutants to identify a number of genes associated with those phenotypes. So he's going to tell us about some of that work today in his talk titled Modulation of Learning and Behavior Selection by the Adaptor Protein 2 Complex. Dr. Jain, whenever you're ready, take it away.
Well, thanks so much for this opportunity to chat about this work that my lab has been investigating, as you said, using larval zebrafish. And this is most of what I'm going to be talking about today is stuff that's not published yet. It's hopefully going to be published soon. We're within probably the final draft of about to submit some of this stuff. So I'd really would appreciate any comments or feedback that folks have to try and improve things. So my lab is really interested in how genes influence behavior, and in particular, how they're involved with regulating learning and how the nervous system can select different behaviors. And the selecting different behaviors is really a critical thing that the nervous system must do. We're sort of surrounded in our environment with all sorts of diverse stimuli, action, movement, sounds, tactile stimuli, things like that, that our nervous system really has to make sense of. And
Dr. Jain, I'm not sure if you wanted to share your slides.
Oh, are they not sharing? Oh,
No.
Sorry about that. Let me figure out. Thanks for letting me know. Let's see. I'm presenting to myself apparently. How here? It says that I was screen sharing.
I could see them.
Oh, was it just me?
Yeah.
Okay.
All right, let me give this a try again.
Okay. Yeah, that's my fault. I figured out what I did wrong. Sorry. I apologize. Go ahead. Sorry to interrupt you.
All right, let's start this up again. All right, can you see the slides now?
Yep, sorry about that. That's my bad.
All right, so we're surrounded in our environment with all sorts of diverse stimuli that we really have to make sense of and choose how to do the appropriate thing for the appropriate scenario, and then adapt to changing environments. Any of you who have been surviving through a pandemic recently know that as you've been working from home, there's lots of stimuli that we need to focus in on or focus away from. So, for example, this woman clearly is focusing on what's really critical, which is getting this phone to the cat that has lost it. But in spite of a variety of other sorts of stimuli. And at the same time, though, she needs to be able to shift her focus and shift what she chooses to do based on what is going on around her and what the current needs are. And so that adaptability is really critical. And we need to be able to set those thresholds of when we do what and how we do the right thing at the right time, so that we don't end up in a scenario where we just completely ignore repetitive stimuli or are hyper responsive to stimuli. And so this is a process that's really crucial for the nervous system under any conditions, but in particular, neuropsychiatric conditions also are known to impact these sorts of abilities. So the ability to learn, to filter out stimuli and decide whether they're relevant or irrelevant, and also to bias their behaviors. And so these sorts of changes are observed in a number of conditions, including autism spectrum, anxiety, addiction, schizophrenia, etc. And what all of these conditions share is that they all have some level of genetic underpinnings, but there's a lot of complexity there. So behaviorally, there's a lot of complexity in terms of, the different sorts of behaviors are regulated in different ways, but also genetically, there's a whole, hundreds of genes that have been implicated in these different conditions. And they all play different sorts of roles here, and it's not entirely clear yet, kind of how they all work together and which ones are regulating different components of these different symptoms. And so our lab is really interested in kind of unpacking the genetics that are underlying these sorts of behavioral choices. And so for the rest of the talk, I'm going to be focusing on our work, as we said, using zebrafish. The first part, I'll be telling you a little bit about how zebrafish, we're using zebrafish to study simple forms of learning and behavior selection. And then for the remainder of the talk, I'm going to focus in on one of the mutants that we pulled out of a genetic screen that's given us a little bit of information on this adaptor protein 2 or AP2 complex, and go into sort of what we found so far about its roles in learning and behavior selection. All right, so the main behavior that we've been focusing on in my lab is the acoustic startle response. And so this is a very simple behavior that's conserved across vertebrates. In humans, you're probably familiar with what this feels like and looks like. Here we have a slow-mo video of an innocent undergrad. And behind her, there is a postdoc lurking there with a balloon. And that postdoc also has a pin in their hand. So you can sort of imagine what's going to happen. So we play our slow-mo video here. We pop the balloon, causing that sudden stimulus. And then we see a very drastic response here, right? So she started off with kind of a protective crouch down, closing her eyes, protecting from a potential threat. And then that's followed up by a sort of a more visceral response that can be more variable from time to time. And so this protective behavior is observed across animals. It's found in zebrafish as well. It looks a little bit different. So here we have a larval zebrafish right there. It's also innocent. It's about 5 days old, so it's free swimming. And we have it in a little petri dish here. And it is being hunted by a hungry dragonfly larva here, right? And so when the dragonfly then attacks, it's going to create a little bit of a ripple and a sound on the water. And you can see it's attacking that fish, and then it performs their startle response to escape from that potential predator and threat. And that in all these cases, this is a very fast response. It's a protective response, and fish, they're often initiating their responses within milliseconds. And so all vertebrates are performing this rapid response, and it's conserved, although it looks somewhat different. It has analogous circuitry that's underlying. And another reason that I've been interested in studying this behavior is that even this simple response can be modulated, and those modulations are shifted in a variety of neuropsychiatric conditions as well. And so when we're studying this in the lab, we're not eliciting this sort of response using dragonfly larvae, but rather we're doing it in a more automated way, where we have our fish in a series of separate wells. And then they're going to be stimulated by acoustic stimuli from a speaker. And then we record at a millisecond resolution what each of those fish are doing in the time before and after those stimuli. So we can really record lots of fish at the same time. Here I'm showing you 16 of them, but we can kind of scale this up as well. And then we can use automated tracking software to analyze the behavior of those fish and categorize exactly what behaviors they're doing, and then what the kinematics of their performance looks like as well. And so, so fish perform this startle response, but they don't just kind of respond in a reflexive way constantly. They can actually filter out irrelevant stimuli that don't have any sort of negative consequences. And so to show you what that looks like, I'll give you a set of fish here where we're giving repeated loud stimuli. They're identical stimuli. We're going to start off by giving those fish stimuli every 20 seconds, and then we're going to continue giving that at a shorter interstimulus interval. And so just to kind of give you a little help in interpreting what's going on with these fish, I've put in little red asterisks that are going to show you the fish that are about to do a startle response. All right, and so you can follow each of these stimuli with this little moving ball down here on the bottom. And as it gets to the middle, right there, that's when the acoustic stimulus is given, right? So when we start off, you can see that the stimulus happens, and then pretty much all those fish are responding in a synchronous way. But then when we give that identical stimulus over and over again, now separated by every second, you can see that the water's still rippling, we're still giving them stimuli, but those fish are becoming much less responsive over that very, very rapid period, all right? But if we give after that, if we give the fish about a couple minutes to recover, they're now all responsive once again, all right? So the fish aren't just getting tired, they're really kind of adapting whether or not they're gonna respond here. And so this process is called habituation, where repeated irrelevant stimuli are going to cause them to dampen the response. And in this case, they stop responding altogether. All right, and so what this shows us is that even in this really simple, very rapid behavior, the fish are showing this short-term habituation learning. And we've done some work previously as I was a postdoc, characterizing this behavior and just really confirming that's a learning process. And so this is great. So this behavior is modulatable, but it's also a great system for studying the selection of behaviors. Because here I've just been showing you, describing this as a single startle response, but fish actually select between two different behaviors when they're confronted with a sudden sound. For the purposes of this talk, I'm going to be calling them the fast and the slow startle behavior. For the aficionados out there, it's in literature, it's called the short latency C start and the long latency C start. But it's, for our purposes, we'll call them the fast and the slow response. They're still both very rapid responses. And so I'm showing you a time series of what the fish are doing over 40 milliseconds after the stimulus. And you can see that in both cases, the fish are turning away from that acoustic stimulus. And then in the fast case, they've actually moved away from their initial position. Whereas with the slow case, they've turned away, but they haven't really gotten too far yet. And so these are actually two distinct behaviors. They're not just kind of a fish reacting quickly and slowly. And so a lot of work has gone into characterizing these two different behaviors. And we've shown that these are distinguishable by their latency and their movement kinematics. The slow response is much more graded, whereas the fast one is sort of an all or nothing response. It does the same thing regardless of what the intensity of the stimulus is. They use different parts of their bodies and different muscles. These slow responses are using their pectoral fins as they start off, whereas the fast responses, they tuck them in before they do anything. And then they're controlled by different neural circuits. A pretty well-known component is this Mauldner neuron, which is the command neuron in the hindbrain. There's a pair of them there that's absolutely essential for this fast response. But you can ablate that neuron and they no longer do fast responses, but they can still do slow responses. And there's a variety of other circuit elements that seem to be distinguishing these two. And then there's also a number of genes that have been identified that that can selectively impact one or the other behavior. One of those from the kind of classic zebrafish screens was called Kindstein, which ablates these slow behaviors but doesn't impact the fast behaviors. So altogether, the fish are actually selecting between these two different behaviors. But what I wanted to first kind of convince myself is that is this actually a relevant selection ethologically? And also, is this selection regulated, or is it just something that's happening relatively randomly? And so ethologically, I was pretty convinced that this is an important decision. So you remember our video that we saw before where that fish did a fast startle response and escaped from their predators. But if they don't choose to do a fast startle response and slow down for dramatic impact here, you can see that these fish responded with a fast startle. This one did not. And so those milliseconds really make a big difference in whether or not fish is going to survive that encounter or whether it's going to be eaten up. All right. And so we wanted to know whether this selection between fast and slow startles is something that's really well regulated. And so to test that, we gave the same fish a variety of stimuli of different intensities. And so what we found is that when we give fish quiet stimuli or low intensity stimuli that are presumably less threatening, the fish are biased towards performing these slow startle responses. Whereas if we give them strong, potentially more threatening stimuli, and they're spaced out by 20 seconds here, they prefer to perform those fast startle responses. However, this isn't really a fixed situation, because now if we give them those same loud stimuli repeatedly and reduce the interval in between them, where I've shown you before that eventually they'll stop responding, before they stop responding, what we see is that they actually shift their behavioral bias. So now those loud threatening stimuli are being perceived as not so threatening, and the fish shift their bias. So they're now, when they respond with those slow responses. All right, so this shows us that these fish are actually dynamically biasing their behavior, and they're able to modulate whether or not to respond in the context of their recent history. All right, so with that system in hand, we then wanted to try and understand, well, what's going on genetically that underlies this? Because these fish are 5 days old. This is really a capacity that their nervous system has already. They don't have to learn how to select between these. So we wanted to figure out what might be underlying that. And so we undertook a forward genetic screen when I was a postdoc to try and identify, based on behavioral relevance, genes that could be involved in modulating this acoustic startle behavior. So I'm not going to go through the whole thing, but it was a multiple generation screen here. And we screened through over 600 different genomes. And through this, identified a number of different classes of mutations that affected acoustic habituation, mutants that affected this behavioral selection between the fast and slow startles, as well as mutants that impacted kinematic performance of the acoustic behaviors. So I'm going to be focusing on one of these mutants for the rest of this talk, which we originally identified as ignorance is bliss and shows a habituation phenotype. So we've got both of these, a sibling on the left and a mutant on the right. They, morphologically, they look pretty similar. And so if we give them just a single loud stimulus, what we see is that both of these fish respond to that stimulus. Oops, what just happened? There we go. Right, both of the fish respond to that stimulus, and their responses look pretty normal. They're responding around the same time. But after repeated stimulation, what we can see is that this mutant is continuing to respond at a time that the sibling has stopped responding to this. All right. So this, showed us that it has a bit of it. This gene was likely to be involved in regulating this habituation process. And so obviously we're very excited to identify what the relevant gene was here. And so through a whole genome sequencing approach, we found that it was a point mutation in a splice site of this gene called AP2S1, which encodes the sigma subunit of the AP2 or adaptor protein 2 complex here. So this is a tetrameric complex, and the sigma subunit is right there. And so this gene has been associated most frequently in humans with a serum ion balance disorder called familial hypocalciuric hypercalcemia type 3, or FHH3. But much more recently, it's been identified to be linked to the autism spectrum disorder, as well as showing learning deficits in children. And so that's been really identified in the last couple of years. And so the mechanisms of those are really not very well understood. So we generated some additional series of alleles of this AP2 sigma gene, or AP2S1, and the other one that I'll point out here is a CRISPR mutation that's a very early truncation, so it's likely to be functionally null. This original allele that we identified, it drops out a single exon, but remains in frame. And so you get this sort of a shortened version of the protein, which actually in humans, this spliceoform is found endogenously, but it hasn't been clear really what the function of it is. So we suspect that this might be a partial loss of function. We're not sure yet. And so this AP2 complex is best known as a complex that regulates clathrin-mediated endocytosis. So the complex interacts with targets on the plasma membrane, and then recruits clathrin to that to allow those targets to be removed from the plasma membrane. And so this functions in a variety of tissues and a variety of times. It's known to be present in the nervous system, so both presynaptically and postsynaptically. So postsynaptically regulating neurotransmitter receptors, but also presynaptically, it can regulate the recycling of the synaptic vesicles. But it's also present in glia and other non-neuronal tissues. And so this FHH3 is actually due to sort of hormonal changes outside of the nervous system. But it's not really clear sort of where and when this gene is actually impacting the behaviors, right? So we wanted to really study that and figure out when and where is it impacting, in particular, this acoustic learning behavior, as well as the behavior selection that we've observed. And then, we also wanted to try and understand, since this is associated with the autism spectrum that has a whole variety of behavioral phenotypes, we wanted to see whether this, in our system, we also could find that it's impacting behaviors outside of the acoustic startle response. So to try and expand on what it's doing with this habituation learning, I've shown you kind of over the course of about 30 stimuli that we have a clear deficit in these AP2S1 mutants, where they can still habituate to some degree, but it's significantly reduced compared to their wild type siblings. And so one of my students, Roshan, wanted to explore this a little bit further and try and figure out, is this just because these fish are sort of slow to learn here? Or does this reflect kind of an absolute cap on their ability to habituate and their capacity for learning here? So what he did was he expanded our habituation assay and went off far beyond 30 stimuli. He gave them, he went out to about 300 stimuli here. And what he found is that the fish do habituate a little bit more if you give them more and more stimuli. But overall, they never reach the same level of this sort of plateau of habituation that the siblings do. And so we modeled and fit the curve of their responsiveness here. And so they are significantly reduced in their total capacity of habituation. But also, if you look at the early stages here, you can see that they have a sort of differential in their speed of habituation learning here too, right? So this the AP2S1 disruptions really impact the rate at which and the number of stimuli that are required to reach their sort of 50% habituation. All right, so together this told us that this complex seems to be regulating both the rate of habituation as well as the total capacity for learning. And so, then we wanted to also explore some of these other components of the modulation of acoustic behavior. And what we found was that the fish actually, when we've disrupted AP2S1, it also disrupts behavior selection. And we hadn't observed this initially when we were performing the screen. But as we gave them stimuli of weak and strong intensity, what we found is that they're totally normal when we give them loud stimuli, but when we drop down to low intensity stimuli, these fish are pretty much always want to perform these fast startle behaviors. They still can perform the slow startles, but they're not up at 100. So it's not that they've kind of ablated that behavior, but just across intensity of stimuli and across contexts, they pretty much prefer these fast startle behaviors, where siblings would normally perform a slow start. All right, and so this is also sort of a partial loss of function here. So we looked at a few of our other alleles, and for all the kind of behavioral things that I'm going to tell you about today, we've looked at a number of our alleles, and all of them are really showing pretty similar behavioral deficits. And so when we looked at our presumptive null allele here, we find a similar sort of shift in their response bias, where they still do perform some of the slow behaviors, but at low intensity stimuli as well as high intensity stimuli, they shift their behavior towards these fast ones. All right, and so this indicates that the AP2 complex does seem to be regulating acoustic escape behavior selection as well as the modulation of learning.
Dr. Jain?
Oh yeah.
I just have there's a quick procedural question in the chat that I think is probably good to ask now rather than wait to the
oh sure, yeah. Please feel
for
free.
the 300 habituation assay. Did you keep the interstimulus interval the same?
Yeah, so that was also at that 1 second interstimulus interval. So we established their baseline with a longer ISI, but we gave them 300 stimuli at 1 second.
Okay. Okay.
Yeah, that's a great question. Yeah, and if there's other questions that come up, please let me know.
Thank you.
All right. So yeah, so this showed us that this complex is affecting both habituation behavior as well as this selection between these two potential behaviors. And oops, here we go. All right, and so although by eye, overall their performance looked pretty normal when we looked at those videos, when we looked a little bit more carefully at the kinematics of their fast and slow startle responses, really we wanted to just kind of make sure that they're actually doing two distinct behaviors, and we weren't getting kind of tricked by sort of a change in their movement performance here. So we looked at a variety of different parameters. We looked at the latency of their escape behavior, sort of the maximum turning angle that they show, as well as the maximum velocity of their escapes. And so if you just look at the siblings here in blue, you can see that there's clear differences between those fast and slow startles, as I mentioned before. So the scale bars here are different on the y-axis, but the fast startle occurs at much shorter latency than the slow startles. And then the kind of the vigor of those slow startles is a bit different. So the angle is lower for the slow startles, and the maximal velocity they reach is also slower. And so when we looked at our mutants, we found that those kinematic parameters are actually shifted significantly, although they're still distinct between those two behaviors. So the mutants actually show they start responding at a shorter latency than usual. You can see right here, and that happens both for the fast and the slow behaviors. But the latencies are still distinct between the fast startle and the slow startle. Then when we look at the turning angle here, we see that the fast behaviors are shifted a little bit weaker angles that they're performing with, whereas the long latency, the slow behaviors are not really changed. And then for the escape velocity, we find that for the fast startles, they're a little bit, they don't reach the same level of maximal velocity, whereas for those slow startle behaviors, they actually are a little bit faster than usual. So what we interpret from this is that this gene seems to be differentially modulating these two different escape behaviors, often in different directions, and somewhat independent of each other. So altogether, we found that that this, this AP2S1 gene, the AP2 sigma, seems to be regulating various aspects of acoustic escape behavior. The habituation, the ability to select between two different escape behaviors, as well as the performance of those behaviors. And so, we were particularly interested to see whether this was reflective of kind of a very subunit specific role for this sigma subunit. So in the case of the FHH disorder in humans, that's been linked to a very specific target that the sigma subunit binds to. And so we were interested to figure out whether this might reflect similarly a specific target of sigma, or whether the entire complex is required. So to try and get at that, we obtained a point mutation in another subunit, the alpha subunit of the complex, and then looked at those fish. They're also viable and look, morphologically normal. And then we looked to see whether those had similar behavioral phenotypes. And so this is work that was done by Rodrigo and Cole in the lab. And so when we looked at the habituation behavior of those AP2A1 or the alpha subunit mutants, we find that they also show a reduction in their ability to habituate compared to their siblings. I'm not going to show this here, but we also looked at their behavioral bias as well as their kinematics, and they actually show very similar behavioral phenotypes to that sigma subunit. And so this suggests that habituation as well as bias and movement performance requires multiple subunits of this complex. And so it's likely reflecting kind of a role of the entire complex. So unfortunately, we don't yet know what the specific target is and which of these subunits might be kind of playing the most salient role. All right, so we've have now a variety of different behavioral deficits or changes when we disrupt different components of the complex, and so we really wanted to get a sense of when and where is this complex doing something to impact this behavior. And so we've taken a transgenic rescue approach to try and figure this out. And so we started off by creating a transgene that paneuronally expresses a GFP tagged version of AP2S1. And then we could express this in the mutant fish as well as their wild type siblings. And so you can see here that this in this larva, it's expressing pretty broadly throughout the brain as well as the spinal cord. And it turns on fairly early in development, about a day or two into development. And so a former student in the lab, Eliza, was looking at this and tried expressing it in our mutant and sibling fish, and then tested them for these various behavioral deficits that we just went through. And so when she looked at the ability to habituate, sort of excitingly, she found that expressing this gene in the nervous system was sufficient to enhance the habituation pretty close to wild type levels. And so we see a significant improvement in their learning here. When then she looked at the bias behavior, and surprisingly, we don't actually see a shift in their bias. So there's potentially a bit of a trend towards shifting their responses back towards these slow responses when we give them weak stimuli, but it's not significant, and we've done this several times. In each case, we haven't found a significant improvement of their behavioral bias selection. So we're seeing a bit of a dissociation in terms of where this gene may be required for these two different modulations of escape behavior. It seems to be affecting habituation within the nervous system, but it seems to be affecting this behavioral bias through a non-neuronal function. So then when we look at the kinematic performance of acoustic behavior, we found sort of another dissociation. So when we look at the latency of their responses, we find no rescue of that by expressing it in the nervous system. Whereas some of these other behavioral features, so the turning angle and the escape velocity, are significantly rescued back to normal levels just by expressing this AP2S1 in the nervous system. So unexpectedly, we found that this complex seems to be regulating independently different kinematic parameters of and this is just focused on the fast escape behavior here. So it seems to be doing that both through neuronal and non-neuronal levels. So then we wanted to try and get at the timing of where this was happening, because our neuronal transgene was expressed pretty much throughout development, as well as while we're providing these acoustic stimuli. So we wanted to try and dissociate whether this was a developmental role in the wiring of the circuitry, or if it was something that was happening acutely to regulate these behaviors. So to address that, Eliza and Emma used a transgene that we generated, which is will allow us to induce expression of that AP2S1 GFP fusion by exposing fish to a brief heat shock. So essentially, we give them a little a 15-minute spa time in the PCR machine at whatever time we want to induce expression. And then we can see robust and ubiquitous expression across the fish just from that 15-minute heat shock, right? And so what Emma and Eliza did was they gave that heat shock at 5 days post-fertilization, so that all of the circuitry to allow the performance of these behaviors, as well as allow the habituation and behavioral selection, has already been developed. At 5 days post-fertilization, they can already make those decisions and they can learn. So she gave them that heat shock and induced expression when they were 5 days old, and then tested their behavior a day later at 6 days. And so again, we looked at both the habituation and behavior selection. And so for all of these fish, I'm showing you fish that are non-transgenic, as well as ones that are carrying the transgene, but all of them have been exposed to that heat shock regimen, just in case that heat shocking itself might impact behavior in some ways. And so what we found is the heat shock by itself didn't really do anything. We still see in the AP2S1 mutants a reduction in their habituation capacity. We also find that they still show that shift in bias towards fast startle behaviors when their siblings prefer slow startles. So then when we induced this transgene and looked at their habituation and behavior selection, what we found is that this acute expression of AP2S1 was sufficient to rescue habituation behavior, but it was not sufficient to rescue this behavioral selection deficit, right? So once again, we're finding a dissociation or a distinction in the timing of this complex and how it regulates these two different modulations of a very simple behavior here, right? So the AP2 sigma seems like it's regulating habituation and behavior selection at different times. Then when we, since we're already on the path of this, we looked at the performance aspects of this fast escape response. And what we found was that unlike with the neural situation, none of those behavioral parameters were rescued by this acute expression of AP2S1. And so that suggested to us that AP2S1 is regulating escape kinematics in a developmental way rather than an acute way. All right, so just to sum up what we found here, because there's a lot of different rescues and non-rescues, when we looked at the neuronal expression of AP2S1, we found that that was sufficient to rescue habituation learning, and it was sufficient to rescue several aspects of escape performance, but not to rescue that shortened latency. It wasn't sufficient to rescue behavior selection. And then when we look at the timing, we find again that acute expression can regulate habituation learning, but it didn't seem to show any ability to rescue either behavior selection or escape performance kinematics. So summing this all up, what we're proposing is that there's actually several different distinguishable mechanisms that the AP2 complex is regulating the different modulations of escape behavior. There seems to be an acute neuronal role by which it regulates habituation learning. And then there seems to be a developmental neuronal role by which it regulates the vigor and the angle and velocity of escape performance. Whereas the behavior selection and latency of their escape performance seems to be regulated at a developmental time point and non-neuronally, so outside of neurons. And so this was kind of surprising to us that even with this very simple behavior, we're really dissociating a variety of different mechanisms that this complex is using to tailor the performance and when and where to do these different behaviors. All right, so we found that at least habituation was regulated at the neuronal level, and so we wanted to try and hone down a little bit more discreetly where it might be functioning to regulate this habituation behavior. And so I mentioned earlier that fast startle behavior is regulated and controlled by a pair of hindbrain neurons called the Meissner neurons. That are absolutely required and critical for initiating that behavior. Because this behavior is initiated within a few milliseconds, it's actually quite a simple circuit. The sound is sensed from the ear, hair cells in the ear, transmitted directly to the hindbrain to those two Meissner neurons, and then the firing of one of those sends their axon directly down the motor, down the spinal cord, where it can directly interact with motor neurons to cause those fish to turn away from the perceived stimuli within milliseconds. So we expressed a fluorescent calcium reporter in those Meissner neurons right here, and so we're showing the cell body of that Meissner neuron. And we can look spatially at that, and we can see both the lateral dendrite here, which is receiving the acoustic excitatory input, as well as the soma of that neuron right there. And what we find is that when we give them a weak stimulus, we can observe sort of an activation from that weak acoustic stimulus at the lateral dendrite. But that's not sufficient to evoke an action potential in this neuron. We don't see the soma lighting up here. And those weak stimuli then are typically not evoking fast startle behaviors. They're evoking slow startles that are independent of the Meissner neuron. Whereas if we give those fish a strong stimulus, what we find is we see that influx in the lateral dendrite, and that now is sufficient to evoke calcium influx throughout the cell body and evoke that fast startle behavior. So we were interested to look at this in our AP2S1 mutants to try and figure out whether there might be a change in the input to that Meissner neuron from the auditory nerve, and also whether there was a change in the activation pattern of the Meissner neuron. And so Christina looked at these, and what she found is that by separating out those two areas, we can see that after habituation, the siblings are no longer firing the Meissner neuron, but the AP2S1 mutants, as expected, they are firing the Meissner neuron regularly, and that behaviorally matches with what we observe. But when she looked at the lateral dendrite, what she found is that there's actually not really much of a difference in terms of the auditory influx and excitation there. If anything, it habituates even more. The mutants are habituating even more than the siblings are. So this suggests that AP2S1 is regulating the firing of the Meissner, but it's independent of the lateral dendrite input. So it's likely to be through some other regulatory mechanisms, regulatory populations that are synapsing on the Meissner neuron. All right. So the escape behavior is regulated not just by acoustic stimuli, but that same circuit can be turned on by a variety of different modalities. So you can elicit those fast startle behaviors through the Meissner neuron through mechanosensory stimuli, which we can evoke by just kind of wiggling a little piezo behind that fish, but also through visual stimuli. So if we have kind of a looming shadow that gets bigger next to the fish, so it looks sort of like the shadow of a predator coming toward it, that can evoke a Meissner-dependent escape behavior to turn away from that spot. And then we can also evoke both fast and slow startle or escape behaviors by sort of chasing the fish around with a dot that kind of follows it around. And so in collaboration with the Orger lab at the Champollion Institute in Lisbon, we took our mutants and we tried to evoke startle behavior from all of these three different modalities to try and figure out whether there's sort of a consistent change in the excitation of the Meissner neuron, whether in all these cases they're sort of hyper responsive and maybe performing these fast startles more than usual. So when we looked at the mechanosensory induced responses that can do both fast and slow startles, surprisingly what we found is that the mutants actually shift their bias towards slow startle behaviors when we give them this mechanic sensory stimulus that's sensed through the lateral line rather than through the ear. And then when we look at the visual stimuli, we find that these looming stimuli just don't evoke fast startle responses at all. They couldn't care less. They just occasionally will move, but they're very slow and long latency movements. They're not these fast startle behaviors here. And when we chase the fish around with a dot, we can sometimes evoke a fast startle response, this sort of fast escape, but a lot of the time it's definitely much reduced compared to their siblings. All right, so what this suggested is that there's even more complexity going on here with this same escape behavior, because it seems to be regulating how and when to do that behavior in very modality specific ways. And so we were particularly focused then on some of these other modalities to see whether maybe this reflects sort of just a deficit in their vision. So to try and test that out, Hannah looked at these fish using a standard visual assay called the optomotor response, where we project stripes underneath those fish and then have those stripes moving either static or moving in a particular direction. And typically what the fish will do is they'll turn and start swimming in the direction of the moving stripes. And it's sort of thought to reflect what happens if a fish is kind of getting swept away by the current. Their environment starts shifting away and they try and stay in the same place by kind of moving in the direction that their environment's moving. And so what Hannah found is that when we look at the wild type and mutant fish between static and moving bars, we find that they can indeed detect that the bars are moving. Those fish will increase their movement when those bars start moving. All right, so showing that they're detecting those moving bars. And then when she looked to see, are they interpreting them properly? Are they swimming in the right direction? They seem to be doing that just fine. They can orient themselves in the correct direction, and if anything, they're actually better at orienting themselves and moving along this visual path. So this suggests that the AP2 complex is not actually required in general for visual responses and for responding to at least some moving stimuli. So there seems to be a difference in terms of which visual stimuli it's impacting the behavioral responses to. And you might notice here that the mutant fish seems like they're not doing a whole lot when the bars are static. And so Hannah looked at what the fish are doing in the absence of any sort of stimuli. And indeed, we find that they're much less spontaneously active, although they still do move, but they move much less than their siblings. And so also in collaboration with the Orger Lab, we then try to quantify exactly what behavior they're doing spontaneously. And so these larvae have kind of a discrete set of different behavior movement patterns that they can do, these different bouts. So we looked at all of them, both in the light and the dark, and we found that the mutant seems to be shifting the behavior patterns that they select to do, even in the absence of stimuli, in the dark as well as in the light. And in both cases, they seem to be shifting towards these kind of less vigorous, weaker sort of stimuli, weaker sort of responses, and away from kind of the faster, stronger responses, which is in contrast to what we found with the acoustic evoked behavior, where the fish are performing and biased towards these fast behaviors. Right. So what this tells us is that the AP2 complex seems to be regulating behavior very broadly across many different contexts and stimulus modalities, and it seems to be shifting the movement selection patterns in a variety of these different cases. All right, so I'm going to stop there and just kind of summarize what I've told you today. So this AP2 complex we found through a forward genetic screen seems to be modulating a variety of different aspects of how organisms can select when and where to do particular behaviors. So it impacts their acoustic learning as well as their behavior selection. I told you about how this mechanistically seems to be functioning and regulating escape behavior through multiple mechanisms that are distinguishable by time as well as their cellular locations. It doesn't seem to be regulating the excitation, the acoustic excitation of those command mouth neurons. So it seems to be functioning outside of that, at least at the level of regulating habituation learning. And then also this complex seems to be broadly regulating behavior selection across a diverse set of contexts. And so moving forward, we're really trying to identify what are the critical regulatory populations of neurons that are regulating the escape behavior and regulating the malneuroactivity, as well as starting to look into the timing and the location of the requirement for this complex in our visual and spontaneous scenarios. And so we're excited to kind of go forward with that. And then also molecularly trying to identify what are those targets of AP2S1 that might be regulating the way that this is shifting behavior. And given that there's a whole variety of mechanisms here, we're anticipating there's probably a number of different molecular targets that are required for these behavioral modulations. So with that, I'd like to thank everybody in my lab across the years. So there's a number of them that have been highlighting along the way that have been really influential in a number of the different components of this, but this has been work that's been happening over the last pretty much 5 years in the lab. As I mentioned, we started this and identified our mutants while I was a postdoc at the University of Pennsylvania in collaboration with Mark Holman and Kurt Marsden. And then we've been, some of our visual and mechanosensory behaviors that we've been looking at have been in collaboration with the Orger Lab and Joao and Alessandro there. And then also, in addition to things that I've told you about today, I have lots of really excited and talented undergrads that have been exploring a variety of other contexts and other aspects of how these and other genes that we got out of our screen are impacting behavior selection and learning. With that, I'll be happy to take any questions that anyone has.
Thanks, Dr. Jain. It's my job to clap on everyone's behalf. Are there questions for Dr. Jain? You could either unmute yourself or raise your hand if it gets too unruly. I guess I can prime the questions maybe by asking a simple one. In the beginning of your talk, when you were describing the habituation phenomenon, you showed some population responses that changed as a function of number of stimulus exposure.
Yeah.
It made me wonder how much variability there was within individuals. Because I could imagine that population response for something like response selection, fast versus slow response selection, could just be driven by separate subgroups that choose one or the other, but it could also be due to individual differences from trial to trial.
Yeah, that's a great
exposure. And I'm wondering if there's neural correlates of that, especially when you look at things like calcium influx.
Yeah, so that's a great question. The short answer is that we haven't, at the neuronal function level, we haven't really looked. We haven't tried to do that sort of correlation where it sounds like you're hinting at like if different individuals in the population may have kind of an inherent bias a little bit shifted towards fast or towards slow startle responses, is that reflected in differences within their neural activity? We haven't looked at that yet. That could be an interesting thing to look at. We know that across different wild type populations, there's clearly different genetic regulation of this because different wild type strains have different kind of baseline biases between the fast and slow startle responses. And they have different kind of rates of habituation as well. And so there's been work from some other labs that have been looking at the individual level where they've just taken populations of wild type fish, sorted them based on their habituation rates. It's sort of like fast and slow habituating populations. And then, raise those up and in-cross them and gone a couple generations. And that's been heritable as well. So there's clearly variation within the population that I think would, is likely to be impacting that as well. because these are not isogenic populations. They're quite
Okay.
polymorphic.
Okay.
Does that answer your question?
It does. It does.
Oh, it looks like there's something in the chat.
Yeah, I'll ask the one that's
Sure.
in the chat from Isabella Green. She's asking, do you think there would be a longer term behavioral change, like over a span of minutes rather than milliseconds, in response to repeated exposure to negatively associated vibration? And then she expands on that. Like if you expose the larvae to repeatedly to the dragonfly, do you think they would exhibit a generalized hyperactivity?
Yeah, so that's an interesting question. So kind of getting at the timing of sort of the learning here, whether it's just at this purely kind of level of seconds of habituation, or whether they're showing a longer term memory deficit or longer term learning deficit. And that's a great question. We don't really see, so for the acoustic startle response, there's not a huge long-term effect of habituation there. There's some contexts where you can see a bit of long-term habituation. If you give them habituating stimuli, let them recover, and then give them another round of it, and then let them recover, they do speed up their habituation. Normal fish will speed up their habituation each time, indicating that they do have some longer-term memory going on there. We haven't explored that with the AP2 sigma mutants yet, but that would be a great thing to look at. But I think the other part of your question is looking at whether if it's not an innocuous stimulus, if there's actually some sort of a negative aspect to that, whether that might impact their kind of what they change their response to there. And you mentioned hyperactivity. So the fish, the AP2 sigma mutants, or any of the mutants that we have in the AP2 complex, they do show hyper responsiveness to weak stimuli, just regardless with our stimuli. So that does seem to be a phenotype. I didn't have time to get into it here, but that's another phenotype that they have. I don't know. I think it would be really interesting to see if we gave them a different sort of stimulus that might actually have a negative consequence to it, and to see whether that shifts, and that might kind of map with the differences in what this complex seems to be doing as it's regulating this escape behavior. So if we look at in the case of those dragonfly larvae, they're getting both a visual, they're probably getting both a visual as well as an acoustic stimulation there. And so I'm not really sure what would happen if we start incorporating multiple stimuli together. And that's actually, I'm proposing a grant to look at that. So I would love to find that out.
Thank you. Thank you. Another question in the chat from Jake Deslauriers. If I'm pronouncing your name wrong, Jake, I'm sorry. Are there any circuit level phenotypes in the AP2 sigma or alpha mutants that might influence the responses to acoustic stimuli? Or do those phenotypes appear to be the result of primarily molecular dysregulation? I guess beyond the calcium stuff that you showed.
Yeah. So that's a great question. We would love to know. And we're in the process right now of looking at looking morphologically at some of the kind of in a candidate approach, looking at some of the neurons that we know are involved with these behaviors. So far, we haven't seen any morphological changes, at least in the Maldner, and we haven't observed any sort of missing components to the circuitry. But as I think you're probably alluding to, we do know that some of these behaviors, so like the habituation behavior, does seem to be acute. It doesn't seem to be a developmental change, whereas some of these things do seem to be developmental, and so we might expect that there could be some wiring changes that are happening there. Right now we're what we need to do is figure out exactly the locations to really study, because it's expressed pretty broadly throughout the nervous system. And so we are taking kind of a whole brain imaging approach that I didn't have time to talk about, where we're using a calcium indicator called Campari to look across the entire brain and see what neural populations are maybe increased in activity or decreased in activity in our mutants, and how that changes when we look at fish that have been trained with repeated stimuli or not. And so we're in the process. We haven't done that with the mutants yet, but with our wild type siblings, we've been able to identify some populations that seem to be shifting their activity in a way that's associated with either habituation or their behavioral bias. And so those are obviously going to be candidate populations to look at both functionally as well as try and figure out what those neurons look like. Are they there? Are they working properly? Are they wired? And how does that wiring match up with the escape circuitry? But we're also, since it seems to be functioning outside of that, we're looking in the visual system as well, because it seems to be pretty heavily enriched in the optic tectum, the optical processing centers of the brain. So we'd expect that it might be doing some stuff there and maybe regulating circuitry there or in the retina.
That's great. And I think that's really important, given, as you pointed out, you were surprised at how complex a role that AP2 sigma has in what appears to be a simple behavioral response. It's really sort of mind-blowing how complex those effects are. So like I think, looking at specific circuits is important. And like Jake has pointed out, and you just answered quite nicely. Another thing that I was sort of wondering about is maybe some of these separate functions could be dissociated if you look at interactions between AP2 sigma and some of the other, some of its target substrates or whatever. So that here's a question. I know that's a question that you can't answer now, but is a strategy to potentially look at specific, residue mutants that will influence interaction with one pathway but not another? Is that something that you're thinking about, or would you rather be focused on neural circuits?
It is something that we're looking at to some degree. So we have another mutation that so there's, in humans, there's a single residue that is associated, the disruptions there are associated with the autism spectrum and learning disabilities, as well as that same residue is also associated with the serum ion balance changes. And so it's not yet clear whether those are connected or not connected. But we have a very small deletion right next to that residue. It's not the residue itself, but like kind of the two residues right next to that. And that shows the same sort of behavioral changes in all of the things that we've been looking at. But we definitely are in the process of trying to kind of swap in the human version that's a missense mutation in that thing, to try and figure out if that critical binding residue that is interacting with certain targets in the sigma subunit might be critical for one or more of these behaviors. Actually, in a whole separate aspect of our lab's research that I didn't get a chance to talk about, another mutant from the screen that doesn't show a learning deficit but does show a shift in their bias is a extracellular calcium sensing receptor, which molecularly interacts with the sigma subunit. So that's a good potential candidate that might be regulating one or more of these behaviors. And so we're in the process of trying to kind of genetically work that out, and then try and figure out if the timing and circuit level requirements for CASR, the calcium sensing receptor, are matching up with the same requirements that we're seeing for AP2S1.
That's great. Wow, that's exciting. That's exciting work, and I think all of it is really, I think it's really important and points to the value of zebrafish at being able to really dissect out these mechanisms so nicely, even and how important that is even for what might appear to be a simple behavior. Yeah,
and I think it's also pretty exciting that it kind of is mapping up, not that we are in any way saying that we have autistic fish here, but I think a lot of the behavioral diversity that's observed in associated with autism, we're finding similar sorts of phenotypes when we look at that here, and similar levels of complexity here. So I think we may be able to try and get a bit of a handle on some of the mechanistic like molecular and circuit level changes that are happening that might underlie some of the phenotypic changes in sort of in humans as well.
Great, great. Well, we are going long, so I don't want to keep you. One of the reasons why we're going long is because I'm really bad at saying goodbye. But I do want to end by saying thank you so much. That was great. As we talked about before we started, we've had a lot of great talks in this seminar series, and I had high expectations for you, and you exceeded those high expectations. It was a great talk. I think it was fascinating, very interesting phenotype, and really interesting phenotype of the mutants as well. It was a lot of fun hearing that story, and I look forward to future chapters of that story, the AP2 Sigma story. So thank you once again. I'll follow up with an email saying thank you again, because like I said, I'm really bad at saying goodbye. But bye for now, and thanks everybody that's still here for coming. Thanks, Dr. Jain.